He is, I initially met Jorge, I think at STB meeting, I guess also at SOCNIS meeting, so I've known him for a while. I think we have shared a lot of things about accessibility in science and disability, being disabled in science. So when I was a grad student here, I broke my leg and I was in a wheelchair, mostly through my postdoc, and navigating that was very difficult. And when I ended up meeting Jorge, I was like, oh, there's someone else that navigates this whole thing. And so I know a lot of students here are interested in going on to teaching and mentoring, and I thought it would be really great to learn from someone. And if you're interested in making a difference in the world, making science more accessible, making laboratories more accessible, and he's going to kind of share those and also his work that he does there.
Yep.
And I think one of the other amazing things he does, he's done lots of outreach and engagement in the Chicago area of engaging both teachers and learning how to make science more accessible and what have you. And I'm very honored and excited to hear your talk. So thank you for coming, Jorge.
Great. Thank you so much for the invitation. All right. Thank you, Anna, and thank you everybody for coming to the talk. This is always the little bit of an awkward part, right? Because I met some of you guys during the graduate lunch, and then some other folks are new faces in the audience, but know that that I'm very excited to visit your beautiful campus and to tell you a little bit about my story as I was coming through from being a little boy in Mundelein, Illinois, to becoming a scientist and a professor and a teacher, and how I got there. Also, a little bit about how I use this device here that I'm using for mobility, my chair. That work? There you go. All right. So to begin my talk, I wanted to talk a little bit about my career path, because I think that it's not a traditional career path, but the more people that I talk to, the more that I realize nobody has a traditional career path. But so in my case, right, so I was born in Illinois, but my parents were both from a place in Mexico called Nuevo León, Mexico. In particular, my family is from Sabinas Hidalgo, which is north of Monterrey. So I got to spend quite a bit of time. I'm the oldest of 3 children, and being the oldest, also being the male, the son, right? A lot of the sort of labor work was given to me all the way through when I was a kid. Basically, as soon as I could like carry a shovel or use a rake, I was out working with my dad. And that's kind of the way that he was raised as well. So he was following along, right? And here is just a few pictures from my childhood. Having fun at the beach here. Here I am with my dad at our house in Mexico. This house they built for as a place where they could retire to, but now that they are retired, they realize all of their grandkids are in Illinois. So they spend less time here than they wish they could. Here I'm also with my grandparents. Yep, and then it seemed like a Tweety bird piñata always showed up at every one of my birthday parties. So this is one of many. So as you can tell probably from the piñata and from the pictures, I am Latino. And the title of this part here is intersection. Sorry that you can't see that completely, but it's intersection. So I took a workshop on trying to identify where we exist in our intersectionality, right? And trying to understand all these different pieces. Turns out we have a lot of labels that we put onto ourselves, but the more of these are so overlapping, right, that somewhere in there is where we find ourselves, right? So that's what I'm going to try to do here and kind of show you guys. Not all these labels I was born with, some of them I was born with, but some of them I either earned later on in life or got them somewhat by surprise. So even though this little boy here looks like he's very curious and going around places, I wasn't actually a very stellar student. And most people would probably say, well, I would tell you that school wasn't for me. I really didn't enjoy it much. Felt like it was kind of like a boring place. I would prefer to be working with my dad or be out in a field somewhere doing something rather than being with my nose stuffed in a book. So when I got to high school, I had really no plans of going to college. And indeed, the last 2 years of high school I spent learn in a vocational college learning how to paint cars and do auto repairs. However, at that same time, I also decided to take up motorcycle racing, and probably wasn't the best idea. Right before my 18th birthday, I got into a motorcycle accident and ended up with a spinal cord injury. And then so I had some new wheels here. And there was quite a bit of work that went into having to adapt my life at that time, not only to be able to make the spaces that I was going into to be more accessible, but also the plans that I had for the rest of my life to be a little bit different. So remember, my last 2 years was spent learning how to do auto collision repair and not how to study or do biology or anything else, right? So I didn't really know what I wanted to do. My senior year of high school, I spent time trying to figure out who I wanted to be. Like everybody does, I guess. So another piece of these intersections that came up then was, of course, my disability. However, it seemed like as soon as I was stuck at home and had to actually poke my nose into a book, I actually started getting good grades, right? And started seeing that, maybe this does kind of work. And I actually graduated and got a certificate, right? To put right next to my sister's honor roll certificates, which it said most improved student, right? And it was like a big confidence booster, and I was like, oh man, like maybe I got it. How far can I take this, right? So after a few years of kind of thinking about what I wanted to do, I spent some time working in a phone bank, right? Or not a phone bank, in a call center, right? Where people would call with their problems, and then I would try to solve them. Their problems were mostly about their dish TV or their direct TV or whatever. But I found out that I was really good at troubleshooting things, right? So I could help you. So even till today, if you guys have a problem with your satellite, my email is at the end. But I really wanted to go in and continue my education. So I thought, how far can I take this? So I went to college, and I went to University of Colorado Springs, and graduated. And in fact, the provost in her speech, she actually mentioned my story, right? Of being like, like here's this student who wasn't sure he was going to go to university, and now he made it all the way through our program, and is even wanting to pursue a research career. So we had this press release, and also like any Latino family that has a person that graduates, right? Everybody comes, right? No, none of these people live in Colorado, right? So everybody came, right? Because they were like, Jorgito graduated. So still though, I thought, what do I do with this, right? So how far can I take this first generation thing? So I'll tell you a story about when I applied to graduate school, because it's probably a story that you guys didn't do. So when I went to university, I said, I'm really smart and I like biology. I want to be a doctor, right? Like a medical doctor. That's what we all say, right? But then when I got to do some research over the summer with the professor, at the end of the research experience, I'm like, I love this stuff. Like, how do I do this? So I went into her office and I said, how do I get your job? And she was like, well, you have to go to graduate school. And I was like, what is graduate school? Is that like medical school? And they're like, no, it's not like medical school. And then she started telling me what it is, right? And I'm like, I think that sounds like that's a good idea. So I went out and I got some lunch and I went out into the quad or whatever, and I like opened my cell phone, right? So this one flipped open. And I looked up the phone number to call Northwestern University. And I like called like the front desk of Northwestern University, right? And I said, so I'm about to graduate from college with a degree in biology and I want to get a PhD. And they're like, why are you calling us? Like, you have to apply. Do you have an application? I'm like, I don't even know what that is. Where do I get it? Do you have a website, right? And then so they shifted me around to all these people. And for some reason, right, like maybe the stars aligned, I don't know what it was, but they got me in contact with a person who was in charge of this program, this collaborative learning and integrated mentoring in the bioscience program, right? Which was one of these first diversity initiatives that they had at Northwestern, right? And like, I didn't know what that was, right? But they were like, wait, so you have a Hispanic sounding name, so we assume, right? Some days. But also you're telling me that you're in a wheelchair? Like, this is a twofer, right? So they're like, we're totally gonna get this grant renewed. This is what I assume is happening, right? And then actually now that I'm on the other side, this probably was happening. And they're like, we gotta get here now, right? So yeah, so they extended an offer and they said, come and visit our campus and tell us what you think. So I came, spent the summer there, and then learned and met all these folks, many of whom I'm still in contact with, and I met, and they were sort of my first cohort and my first set of fellow scientists. But the truth is that I went to graduate school and I wanted to go to medical school and I was interested in biology, not because I had some intrinsic interest in biology, right? I wasn't just trying to figure the innermost secrets of life out. My plan was to cure spinal cord injury, right? I was like, this one thing that destroyed my plans, right? How do I make that not happen anymore, right? Or how do I repair it, right? I had spent a year and a half in physical therapy trying to relearn how to walk again, right? Not able to. They put me in these like in these contraptions and all these things. Maybe some of you guys work on them, right? And I remember one night in the hospital that I had heard about Christopher Reeve, right? And they were like, they put him in this machine and now he can walk, right? And I was like, no way, like that can't be true. And I like went and I like looked for the TV, I looked for the TV show, right? So I could watch, so I could see it. And then I saw it and I was like, that's not walking, right? They had him with all these straps on in this machine that was walking for him. I'm like, that's not what I imagined, right? A cure for spinal cord injury, right? A robot that can pick you up and then take you places, just an advanced chair, right? Sure, it'd be great if one could take you up steps, maybe even that step there, right? But that's not a cure. And I remember also speaking to my own neurosurgeon, right? When I was after my spinal cord injury, immediately after I couldn't feel my legs, right? And I said, I can't feel my legs. And I remember some Boy Scout outing or something where they told me, like, if you ever fall and you can't feel your legs, don't move, right? And I don't know why that stuck in my mind or why it like revealed itself at that moment, but I'm like, oh, I fell and I can't feel my legs, I should not move, right? But in my mind, as a mechanic, right, as an auto mechanic, as a person that repairs cars that have gotten into collision, I thought all they have to do is get me to a doctor, right? They get me to the right doctor and the doctor can take those parts out that are broken and put in the fixed. So when the doctor came into the office, into my room, and I saw him, and I had gone through my morphine, whatever, withdrawals, and I said, fine, doctor, like you can fix me, right? Like you can fix. And he said to me, oh, we haven't figured that one out yet. Right? And I thought, you haven't figured this one out yet? Like, we put a person on the moon. We put a person on the moon, and I fell off a bike. Right? You can't put me back together, but you can put somebody on the moon. Doesn't make any sense. Right? So all of that was percolating, right? And I come into graduate school and I think I come into that room and I say, I'm gonna cure spinal cord injury. That's what I'm here to do, right? So maybe I don't feel like I was the best prepared person to be there, right? I would look at my classmates and I would say, man, they went to these private schools, they had tutors, they had everything else, right? Me, most of my formative years were spent at landscaping with my dad. How am I going to compete with them? But now I had a mission, right? I said, I'm going to cure spinal cord injury, and I'm here. So then I met Jacek. Jacek was a developmental biologist, and he taught my developmental neurobiology class. And after class, he's like, you're asking a lot of questions. Because I was sitting in front. My hand was up all the time. What are you here to do? I think I asked some of the graduate students this earlier today. What are you here to do? And I said, well, I'm here to cure spinal cord injury. He's like, how do you know how to repair a spinal cord injury? We don't know how one is built in the first place. Right? We don't know how a spinal cord is built in the first place. How are we going to learn how we can repair one that's been injured? And I was like, right? Like that was one of the moments where something all of a sudden clicked. And I said, wow, how do I learn how to build a spinal cord? He was trying to get me into his lab. Right? Because the next thing he said was like, you know what? I think, actually, how Jacek did, I think I have the perfect project for you. And I said, really? What do you do? He said, well, I study these things called zebrafish. And I'm like, that has nothing to do with human biology. And he said, no, but they do. So he introduced me to these little guys, who some of you all work with. These are zebrafish. There's a nice little female running by there. There's a male, right? So these guys here, these are our zebrafish. And I was like, well, that's interesting, but so what if I cut a spinal cord on a zebrafish, it'll regrow? And he's like, not necessarily. And I said, okay, so how is this going to help me? And he's like, well, we have to look a little bit younger. Right? We have to look a little bit younger. And what's interesting about these fish is that if we set up two fish, this male and female fish, inside of one of these, which we call a crossing tank. Some of you guys have seen them before. And we leave them overnight, we play some romantic music. And then in the morning, what happens as soon as the lights turn on, they start to interact with each other. And then they lay eggs, and the eggs kind of fall down here, in this area here, right? And you have access to these zebrafish embryos from the single cell stage, right? And at the time, I'm like, what does a single cell have to do with the spinal cord, right? Like, I don't want embryon, I don't need a single cell. I need a spinal cord. And he's like, yeah, but look, in 24 hours, you have this, right? In 24 hours, you have this. You have a little fish inside of a shell. And guess what? It moves. It wiggles its tail, right? And what does that mean? What do I care if it wiggles its tail or not? Well, if it wiggles its tail, means that there must be something wiggling that tail, right? It must have nerves that are connected that are wiggling that tail, right? So I was like, okay, all right. I think you got me, Jacek. I think you got me. And he's like, no, not yet. Hold on. I want you to see this happen because you can watch it happen under the microscope. And he showed me this video, right? And this video, probably everybody who's ever studied development has probably watched this video. But I think that we should take a moment and just appreciate the beauty of the zebrafish embryo as it's developing, right? So here's the yolk ball, which will come into importance in a moment. Here's the actual animal breast. And you see it dividing. Eventually it becomes this blastula, right? Then you go through the gastrulation phase. So I know you guys are like, this isn't supposed to be developmental biology. So then, you get convergence and extension, right? See, so I can pass my prelims. So we get our eye right here, right? Here's the eye field, right? Here's the somites, here's the tail bud. The yolk makes a beautiful change, right? There you go.
So that's 18 hours, a little bit less than 18 hours of zebrafish development. But Jacek said he had a project for me.
He said, I have a project for you. It's very interesting. And he showed me these pictures. These are the exact pictures that he showed me, right? When he was trying to recruit me to his lab. It's like these pictures, and they're of an in situ hybridization. Some people call it in situ. Jacek calls it in situ. So that's what I call it. So here the yolk has been removed, but this zebrafish here, so this is a 24-hour-old zebrafish, 1 day post-fertilization is what that stands for. And it's stained with an antisense probe against a gene called NODM2. And what's interesting is that the gene is expressed along this area of the fish, which is called the horizontal myocephalon. Right? And it's where the two edges of the chevron of the somite kind of meet, these muscles here. In a set of muscles called the muscle pioneers. Here's a better shot over here. And this is where I started to fall in love, not just with zebrafish, but also with these expression patterns, right? So if you could see where the gene is being expressed, right, maybe you can kind of start to develop hypotheses about what role it might be playing in development, right? Or what role it might be playing in cellular physiology. So for example, we have a gene here that's being expressed inside of a muscle tissue, right? In a very specific, discrete spot in the trunk of the fish that is actually used as a landmark for neuron outgrowth. But I remembered that video that he showed me, right, of the fish developing, 1 cell, 2 cell, 4 cells, right? And I said, but like static images are one thing, but a video, right, is something else entirely, right? You can see so much, right? If a picture is worth 1,000 words, imagine what a video is worth. So I said, I want to see these neurons growing, right? And see how they're interacting with these cells are muscle pioneers. One of the fish that he had in the lab that I think just needed to be crossed, and nobody was maintaining them, right, was this fish called the Olig-2 GFP fish. And this fish has green fluorescent protein being expressed under the promoter for the Olig-2 gene, which at this stage of development is expressed in motor neurons and some interneurons.
And I use confocal microscopy to image these neurons growing.
And you can see that at the beginning of the video, the neuron had already been extended from the spinal cord, but up here is the spinal cord, right? And then here's that chevron shape, if you can imagine the muscle has been removed from the picture. You can see this neuron grows out of the spinal cord, makes its connection, and then goes on down, and then innervates the bottommost of the myosome. And to a kid that's trying to cure spinal cord injury, right? You think, oh, wait a second, I can watch these neurons grow, right? So I can reconnect neuron. Amazing. This is exactly what I want to do. It's not that easy.
But to me, at that time, hell yeah, yes, it's that easy. Let's keep doing it. It takes about 3 years for that to wear off, right? My 3-year folks, right? Okay. All right. So we'll watch it one more time because it's just amazing. Plus, it's probably my favorite video. You see the growth cone here growing out. Innervates the bottommost. And all along the way, it's making connections and plus on. But that's for a different talk. Okay. So together with Jacek and Jacek's lab, I was able to demonstrate that gene that was expressed in these motor neurons could actually play a role in motor neuron guidance, in motor axon guidance,
And we were able to publish a paper, describing its role in primary motor neuron outgrowth and innervation. But more importantly, right, was that here I was demonstrating to the scientific community that I was worthy of being a scientist, right? I was like, so one of the questions that I asked the students that I had lunch with today was, how many of you students consider yourselves scientists? So maybe in this hall right here, I could ask this person, how many of you all consider yourself a scientist? I think that's everybody. I must be in the right building. I didn't, right? I did not consider myself a scientist. And some people might have thought, oh, okay, so this is that imposter syndrome thing, right? But no, I don't think it was that. I think I was straight up an imposter, right? Like I'm like, I came in here and somebody messed up, right? Like I'm like, I came in with enough confidence that people were like, oh, you know what he's doing. I didn't know he knew what I was doing, right? But I had the drive, right? Because I had something that I wanted to complete. I wanted to cure spinal cord injury, right? So now I just needed somebody to say, okay, you're a scientist. And if I'm a scientist, okay, so then now I can get a lab, right? And if I can get a lab, then I can start really working on this problem of spinal cord injury. So I published this paper. I was able to convince my thesis committee that in fact, yes, I am a scientist, right? I'm glad that everybody here considers themselves scientists. But if you don't, that's okay. You'll get there. And so here's me, Jacek, on a very happy day when I was able to get my PhD. So now I could consider myself, or I thought I could consider myself a scientist, right? But then something happened, right? So my wife and I, due to my injury, right? Things are a little bit more complicated when you're trying to have babies. Things have to be planned out, right? There's some clinics involved in a very awkward office visit. But in so doing, what ended up happening here was that we transferred 2 embryos, and I'm a scientist. I say, oh, I transferred 2 embryos. I'm going to have twins. This is awesome, right? And then all of a sudden, they call us in and they're like, I just want you to watch the video. And you want to film, like we're going to share something. And then okay, here's baby A. That's the heartbeat. Here's baby B. And here's baby C. They're like, what is that? Wait, what? There was 2 embryos. How did it 3 babies? But yeah, but for sure. So here, this is either Edward and Enrique. This is Edward or Enrique, and it's frozen. I know that. So I had these 3 kiddos, and then now they decide to tell me, right? So now I'm a dad, right? And now they decide to tell me, oh yeah, no, you got your PhD, right? There's this thing called a postdoc, right? And I was like, wait a second. When I went into Dr. Kimball's office, right? When I was an undergrad, she told me how to get her job. She didn't tell me there was a postdoc. She just had graduate school, right? I should ask more questions. But anyway, so now, there's a postdoc that may or may never end, right? So I started looking for jobs, right? So I'm like, I got 3 mouths to feed, 4 mouths, I guess I have to feed my wife. And, I never have a problem. So now I had to find a job, right? So now kind of these dreams, right? These ideas of having this R1 lab, of this research, of this research lab starts to kind of fade, right? And I'm like, okay, well, hold on a second. So I got to rethink my priorities again. I had to rethink my priorities when I wanted to be an auto mechanic. Now I want to be a scientist studying spinal cord injury. And now I have to rethink my priorities here, right? I have to make sure that everybody's taken care of. So I ended up finding a job in a small, primarily undergraduate institution called Northeastern Illinois University, where I'm still now. I would have taken any job, right? Like I was pretty desperate, right? So they were like, okay, so I got a degree in neurobiology. They're like, sorry, we need like a cell biologist. And I'm like, a neuron is a cell. Right? Yeah, I can teach cell biology. Yeah, but we need you to be able to teach anatomy and physiology. I'm like, I'll get up on that table with a cadaver. Like, you just give me the saw, I'll take care of it. Whatever it is. Right? So finally I was able to, finagle them enough to convince them to give me a job. And my wife took a picture of all her kids on their first day of school, so she took one of the first day of the class. Right? And then this was my lab space, right? So they didn't have very much lab space. I think my startup was like less than $20,000. So that paid for one microscope. And this space here, which is basically just a room in the basement, right? But I thought, this is mine, right? This is mine. This is where I'm going to cure spinal cord injury. That thought is still in my mind. Also, with the new lab comes students, right? So I'm like, okay, so I have this lab. I need somebody to do the experiments with me and to reach things that are on high shelves. So I recruited my first group of students for that summer. So I brought together some students. And then in addition to students, we needed fish. So I had these 2 tanks. So I had 2 20-liter tanks. And then went to Yasik and asked for some fish. Gave him some fish, some wild tanks. And I put them in here and I started getting to work.
In addition to that, in addition to the stuff that I did in my lab, right? The other part of my job, the thing that was actually paying my salary was my time as a teacher.
And when you are in an institute like Northeastern, which is a very small school with a small budget, small research budget, you have to find clever ways of doing your research. One of the ways that I found was incorporating my research into my classes, right? So I'm like, oh, this way I can put it into lab fees or whatever. And then I can pay for some reagents. At the same time, I could give students, some research experience by incorporating it into class. So that's what I did. So I thought, well, if I'm going to do this in a classroom, I don't have any fancy microscopes, I don't have any fancy tools, right? I need something that's very easy for students to visualize and count. And, I want them to have a hypothesis. I want them to be able to collect data, right? How do I do this? So I thought back, like, how did Yasir get me? The picture of the gene expression, the picture of the in situ, right? So let me take my students and we'll identify some genes that have interesting expression patterns and then try to study them further, right? As part of my cell biology class. Remember, the neuron is just that, right? So that's what we did. So as students started looking into gene expression patterns, this is one of my favorite websites ever, ZFin. Sure, everybody, well, if you're in ZFish, you've heard of it, but they have a great button that's called expression. And if you click on it and you type in some anatomical term, they'll show you all the genes that are expressed in that place with some beautiful pictures by the Thies Lab. So I typed in something or another, and then students are very clever in this way, right? Because I'm thinking to myself, I'm like, oh, neurons, yeah. Like, we just want to look at neurons. The students are like, yeah, but there's other stuff there, other stuff that's interesting. So one of them thought, what about the yolk, right? I'm like, the yolk. The thing that I remove when I take pictures of things? And they're like, yeah, the yolk, what does it do? It's actually quite interesting, right? So we looked at expression patterns of the yolk, right? And I looked at it and I said, wow, that's easy to see. You don't need a fancy microscope in order to see the yolk, right? It's measurable, right? You can take a ruler and actually put it from one side to the other and actually see how big a yolk is, right? It's a source of nutrients for the fish. What else do I know about this yolk, right? And it's got beautiful expression patterns of genes that are expressed just in the yolk, right? So we have tissue-specific expression of different genes.
right? Nobody's studying it, right? So I think it's great. So I took students and I started looking at the yolk and just trying to monitor how does the yolk change over time. And just to see if we can observe some pattern of absorption, right? And what you see is that the yolk, of course, at early stages, so all of these are at the same scale, but you can see that the yolk here at the early blastula stage, right? Here's the yolk at 36 hours, here's the yolk at 4 days, here's the yolk at 6 days, right? And start to get smaller over time. And you can track this as it gets smaller, right? And see how things affect the absorption of that yolk. Right? As I looked into it a little bit deeper, right? You start to identify different things within the yolk. It's not just as simple as being a yolk cell, right? There's also this beautiful yolk syncytial layer. So these are nuclei from cells that have actually dropped into the yolk, right? And then there's this beautiful cytoskeletal a network there that's moving around those nuclei all around the yolk, right? So that they completely surround it, so that by this stage, you have all these nuclei surrounding the yolk. Remember, I showed you that it changes, it morphs like this. In addition, if I use some fluorescent stain for lysosomes, you end up seeing that there's actually this yolk, what we call the cortical lysosome network, that's all around this cortex of the yolk. And that's what's helping to bring in nutrients from the yolk and digest. So here's an example of that. So here we use, I believe this is Curtin orange, right, to monitor these. Sorry, no, this is just lysotracker, and to monitor the development of lysosomes throughout this. And what you can see is this cortical lysosome network is not there originally, but as this fish develops, at least 18 hours, start to see it develop. So I started to look at different genes that are expressed during the yolk, during different stages of development, specifically focusing on some of those lysosomal genes, in particular the mTOR pathways. And what we noticed was that there's dynamic changes in the expression of certain of these genes from when they're forming the yolk. So before the yolk syncytial layer forms, yolk syncytial layer formation, during the process of yolk absorption, and then lastly, in this time during free feeding. So this is where the fish stops dependent on the yolk nutrients, starts actually finding paramecium. Right? So we see this change in expression pattern, right? So we started looking at these individual genes and started to identify ones that would be interesting to see if they could affect yolk absorption. Before we did any of that, because what we found was many of those genes were associated with this mTOR signaling pathway. The mTOR signaling pathway stands for the target of rapamycin. So when you bathe the fish in rapamycin, you actually inhibit the TOR signaling pathway. The TOR signaling pathway you can think of as the gas in the brake for nutrient absorption. So like when it's activated, it absorbs nutrients, and then when the brake is activated, then you prevent nutrient absorption. So here you can activate the brake by putting in rapamycin, and you can see that the yolk does not get absorbed fully. So we thought, can we recreate this phenotype by injecting, by knocking down certain genes that we find in our genetic screen? Sorry, this went way too sciencey than I wanted it to. Sorry. So here's where we track just the size of the yolk and then compare that to rapamycin, right? You can see that yolk absorption is delayed when you inhibit the TOR signaling pathway. And then also at the time we were learning this fancy new technique called CRISPR, right? And we were reaching out to different labs to figure out easy, cheap ways to make guide RNAs to inhibit, to target the Cas9 protein in different areas of the genome. And so we found this process here, which is beautifully explained in the Moravec Pellegrini chapter that we use for making this no clone synthesis of single guide RNAs. We injected it directly into the yolk cell at about 3 days post-fertilization,
And I want to remind you that I'm having a whole group of undergrads do this, right? So like we identified these genes and I was like, you know, we found these genes, what do you guys think they're gonna do? And we're all developing hypotheses, we're doing this. And then the students were like, well, if we inject it after the yolks and tissue layers form, then it should be cell autonomous, right? It should only affect the yolk, right? So if you affect the mTOR signaling pathway in the rest of the fish, you know, you might have other effects that are not directly related to the yolk. So instead, we injected things directly into the yolk, affecting only the yolk. And in this case, what we used was this enzyme dead version of Cas9 nuclease, which is more of a knockdown technique rather than it doesn't cause mutations. And then students just used a basic grid like this to score the size of the different yolk balls. Don't get your hopes up. I had them present in groups. There were 6 groups. They all presented their own gene. None of them did anything. Except for one. There always has to be one, right? Except for one. Not that one. This one, okay? All right, except for this one, right? So this one is a gene called ADIC, right? And ADIC had the exact opposite effect of what we thought it would, right? So we were looking for genes that would prevent yolk absorption. And what we ended up doing was accelerating yolk absorption when we injected this into the fish. And then so we have to repeat these experiments, right? Because again, this was done. Oh, well, so I did the microinjections because you know, they required a little bit of that. You know, we need to repeat these experiments just to verify that this phenotype is real. But in the data, we followed their yolk absorption over time. I'm sorry, unaccelerated yolk absorption. But even though we didn't find our Nature paper gene, right? What I did was I had about 24 students, right? All took part in these experiments. And they all learned how to set up an experiment. They all learned how to make these guide RNAs, how to work with CRISPR, how to work with fish, learned about yolk absorption. We had the most interesting conversations with each other. We had fun, right? This was an entire summer of work, right? And I didn't think about spinal cord injury even once, right? Working with these students, they started to show me, right, a little bit something else about myself beyond just being a scientist, right? Because what they attached themselves to, right, was the fact that, you know, I was a first generation student. They were all first generations. I was Latino. Probably about 40% of the class was Latino. I had a disability, right? And they would see me and they would see my obvious disability, right? But they would see that I was still doing it, that I was driven, right? Driven to do this. In addition to that, I'm very open with my class. So they know everything about me. They know my kids, right? My kids sometimes would come with me to the lab, right? So they know that I'm a dad, right? They know, you know, some of my ideas about considering myself a scientist, how I want them to consider them a scientist. I asked them at the beginning of the class, how many of you feel like biologists? Who would call themselves a biologist? And you'd be surprised by how few people raise their hand in a cell biology class. But by the end of a class like this, I asked them, how many of you feel like biologists? And just like you guys did, everybody raised their hand, right? Imagine capturing that early in your career. We all lived through the pandemic. We all, hopefully, or some of us got to experience teaching on Zoom. Some of us got to experience learning on Zoom. My daughter, Rosalie, she has cerebral palsy. And we spent the entire time on Zoom, where my wife and I, more my wife, was her physical therapist, her teacher, right, her occupational therapist, all of these things, right? Me, I would come in for the fun part, right? So when it was story time, that's when I would show up, right, on Zoom, and be like, time to read some stories, right? Or I was there for the hard part. One of the things that I love doing though, is watching movies with my daughter, right? Because she has a set schedule of movies, right? Like she's like, okay, first we watch Moana, right? Moana's always first. Then we go to Frozen, right? And then we put in Trolls World Tour just to like lighten it up. And then we go to Frozen 2, right? We need to know how it ends. And then like after that, maybe you'll get Ferdinand, right? So then when this new movie came out, Soul, I was like, Rosie, we got Soul. Can we watch it? So the way that she communicates is she says yes and no, right? So you put your hands out and say, Rosie, do you want to watch Soul? Yes or no? No. You want to watch Soul? Yes. No. So then I just, yes. And she didn't have a choice, right? So we watched this movie called Soul. How many folks have seen Soul? Oh, beautiful. Okay, great. All right. So I'll recap just this part for you. So this fella here, right? He's a jazz musician that has become a teacher, right? Sounds a little good. So he's a jazz musician that has become a teacher, but he always wanted to be a jazz musician. He tries out and gets into a jazz quartet, right? With this woman here, Dorothea Williams. And they go and they play an awesome set together for the first time, right? And he's gone through this entire charade in order to be able to play, right? And then they come out onto the street, right? And then she looks over at him and looks real sad. He's like, what do we do next? She's like, well, we'll play it again tomorrow. She's like, why do you look so upset? And he says, I just thought I would feel different when I got here. And she says, let me tell you a story about a fish. Says, there was an old fish and there was a young fish. The young fish goes up to the old fish and says, I'm looking for this thing called the ocean. The ocean? You're looking at it. Says, what, this? This is just water. Right? I started falling. Right? And Rose is watching this and she's like, there's no singing. This guy crying. This guy's responsible for me, right? I was like, my God, like that spoke so cleanly to me, right? That I had gone into graduate school, right, with the idea that I would cure spinal fluid. And slowly but surely, my priorities changed, right? My ideas changed. Things were not as easy as you thought they were, right? Maybe things were not as important as you thought they were. What was important, what wasn't important. We learned a lot over the pandemic, what is important, what is not important. But all along the way, we've been learning. Maybe it was just more clear with that. And this, for whatever reason, spoke to me so clearly, right? That I'm trying to find this thing called the ocean, right? But really, I think that I'd already been swimming in it the whole time. So I went back to the lab, right? And the lab has been updated a little bit now. So instead of just having 2 tanks, now we have 6, 7, 8. Here's the 2 originals down here. Here's another one here, right? I also have students that are working in there every week, right? And these students are going out to do amazing things, right? Like they're going out and they're going on to become, some of them have gone to PA school, right? Some of them have gone on to be pharmacologists. Some of them have gone on to be genetic counselors, a COVID nurse, right? Like all these students that have gone through my lab and who, if they had not come into my lab, I don't know where they would be, right? I'm sure they're smart enough, they would have done something really well, but I feel like so happy to have been able to help them.
they still help me reach things that are up high. Right? Which is very important, right? Because especially when you're in a small space, you need that shelf space. Oh yeah, we also have one of these reachers, right? I think everybody should have one of these no matter what, like, you know. I got tenure, right? That should give me an applause. I think I tried hard for that one. All right. I finally got a fish rack. Right. So I got a fish rack now, so I can start to build out a little bit. And also, my class showed me another expression pattern. All right. So along in our little gene expression soirees, we identified this gene that was an uncharacterized gene. All right. Didn't even have a name attached to it. But immediately I thought, wow, this is very cool expression pattern, right? One, it's expressed in neurons, right? Remember, a neuron is just a cell. Expressed in neurons. And in particular, it's expressing these neurons in the trigeminal ganglion and the dorsal neurons here. These are primarily sensory neurons, right? And this beautiful pattern. And this is an uncharacterized gene. Like, when do you find a gene that doesn't have a name, right? What CB979, something like this, like it was just a number attached to it. Right. So I took the sequence and I took a closer look at it, and it has very high similarity to a gene in humans called PIG11, or tumor protein 53 induced gene 11, or which my lab more affectionately calls it PIG11. Right? And PIG11 has two copies of deep fish, PIG11A and PIG11B. It has high conservation, right, of these highly similar sequences, amino acid sequence. Right. And when you look at the data that they have in cancer cells, what they show is that in cancer cells, this PIG11 gene appears to inhibit cellular proliferation and promotes apoptosis. Right? So in particular, in cells that have detached from the bottom of the well, right? So we use the cell culture, knows that there's adherent cell lines, and usually when those adherent cell lines come off the bottom of the well and they start to float, they will undergo apoptosis, right? Because they've lost that extracellular matrix anchorage, right? So when you lose that, you get that. So I thought, well, that's pretty interesting because this is expressed in the Rohan beard neurons, right? So we did some work taking a closer look at where this expression pattern is, and it's expressed in these cells called the Rohan beard neurons, which undergo developmentally regulated apoptosis over time. Here's another view of the Rohan beard neurons. And there's this set of neurons, these sensory neurons, that are sort of transient during development of the fish. And it's what's involved in that response to the touch response, the touch and escape response. All right. And like I said, Rohan beard neurons undergo programmed cell death. So over several days, what you end up getting is an increase in the number of apoptotic neurons. And if we follow PIG11 expression over time, you see that it also decreases. So at 24 hours, we have a high number of positive cells here, and over time, we have a decrease in the number. So either there's less Rohan beard neurons at this time to express PIG11, or somehow this PIG11 gene may be promoting their apoptosis. So we made a knockout using CRISPR-Cas9, and we targeted a Cas9 mutation right to the to exon 3 to disrupt the start codon, and we ended up with a fish. So here's our wild type fish that I got show you guys earlier, and this is how they swim usually. Now here's what happens. These guys are the F2 generation. These injected fish. And you can see that they have this very strange swimming pattern, which my student Christina named the drowning fish. we've had a heck of a time trying to sequence this mutation, but we have finally gotten to the point where we've actually found where the mutation is, and it's an insertion not at the start codon, but actually just 5' to it. So there's this insertion here that disrupts not in the coding region, but in the 5' region, which we're not sure how this, right, relates to the effect that we're seeing, whether this is an effect of P11 itself, right, or whether it's changing the levels of P11. So the next experiments that we're doing are to identify, use QR to PCR to identify the levels of P11. All right. I'm going to go through these last couple slides rather quickly. So we have this knockout, right, that has this weird drowning phenotype. So we thought, does P11 promote apoptosis in zebrafish cell? So it's kind of like, you know, one of those experiments that you're like, well, let's just get everything at once, right, and see if it causes apoptosis anywhere. So we made one of these plasmids with a heat shock inducible promoter driving expression of P11 and a GFP marker. And our plan was to inject the plasmid, induce heat shock, and then monitor the cells that express P11 and see if they undergo apoptosis, either using immunostaining or live staining. And when we looked at the number of GFP positive cells and the number of apoptotic cells, we see that we do not see an increase in apoptotic cells, even in those that are expressing our P11 protein. So it doesn't seem to cause apoptosis everywhere. And the next experiments that we're doing is to try to drive this expression specifically in the neuron. So it leaves us with a lot of open questions, which I am actively recruiting undergraduate students. The thing about undergraduate students is they come, you train them, and then they graduate, right? So it's kind of hard to like have a continuous motion going. But I do want to highlight the 3 students that have done quite a lot of the work that I presented today. One is Cristina Rojas. She's currently at a postbac in Stanford. We have Christian Torres, and he's currently looking for a postbac as well. I believe he should hear back this week. And then my newest student is Ala Ruiz, and she's currently working on the P11 project. My acknowledgements are my funding, which a lot of my funding comes from the Student Center for Science Engagement, the Chicago Checker Cancer Health Equity Coalition, U.S. Department of Education, Society for Developmental Biology supported Christian's work. He was a SDB Choose Development Fellow. And then, of course, it's really hard to get these kids all together in a picture these days. So this is the only one that I could find. This is either Edward and Enrique, and this is Rosalie. Any questions?
Any questions for me? I have a question about, does the precocious loss of yolk cause any spinal cord development issues?
Oh, you know, I don't think so. So the problem with the yolk phenotypes we've seen so far is that the fish don't really make it past these 4 days of development. At least the ones that have the effect. But that is something that we.
The right number?
The right number of somites?
Yeah.
You know, I don't know. Yeah, it's an interesting question. I'll have to take a look at it. I haven't had somebody pick back up the yolk.
Yeah.
Right? Because like as excited as I am about it, students are like, yo, right? But what's super interesting is that, you know, the yolk has a lot of this protein called vitellogenin in it. And fish and other egg-laying animals have 3 copies of vitellogenin, VTG1, 2, and 3, right? And if you look at our genome, we have remnants of VTG as well, right? So it's an interesting gene to study evolutionarily.
So the gene that caused accelerated yolk absorption did the opposite of what you're expected to do when you knocked it out. Do you think that was due to like some type of genetic compensation that happened?
You know, I don't know, but that is definitely a good question. Now, the technique that we use to knock down the gene was using this enzyme dead version of Cas9, where I have seen this genetic compensation happening is when you actually cause a mutation. But I think the data that showed no compensation was when they used morpholina, if I remember correctly. So that's a different antisense oligonucleotide. But definitely it could be happening.
Also very good talk.
Yeah. Thanks.
Right.
I was really intrigued by your approach of just asking students at different times when they feel like, or not when, whether they identify as biologists.
Yeah.
And I wonder if you could say more about, I mean, I suppose it's different for different students, but how/when, how do you think that light bulb goes on? What are they doing when it happens, right? If you could imagine asking them, sort of making a video, continuously, at some point there's a switch.
Yeah.
Do you have any idea what governs that inflection point and how we can understand it?
I don't, and I do have some IRB in the works. To try to actually test this exact question, because it was something that I kind of did kind of by accident, right? Because so none of my classes are more than 24 students. So I was having small class sizes, even in general biology. And sometimes I would have the same students in general biology that I would have in neurobiology, and then I would have them again, sometimes even in my graduate class, right? So I've had these students throughout this time. And I've seen them, it's kind of like a time series, right? You're able to watch them mature as they're going through this process. And I would love to know exactly when that switch happens, right? Of when they start thinking of themselves as biologists. In particular, I would like to know the difference in that between those folks who are from underrepresented groups and how it compares to for other folks, and see what that difference is, right? Because I definitely, throughout a lot of my time, even when I was in graduate school, I would confidently say, I feel like science, right? And it wasn't until a while to say, well, now I think experimental science. Well, why did I feel that way? Like I should have, from the moment you do an experiment and you figure out you need a control, I think you're a scientist, right? So it's a difference between playing in a sandbox, right, and engineering a sandcastle, right? Yeah, but it is something that I'm looking into. It's been my head right now.
To that, thinking about this like in your study chart, maybe you have as a URM, like I know that this comes up, like I can identify with you, I feel like the door's open, it's easier to, I haven't thought about like reflecting on that, not only on the student, but who the professor is.
Yeah.
What that background is and like making those comparisons, break down potentially some barriers that might exist.
Exactly. No, I think that's definitely the case, and one of the things that we're working on is we're developing a professional learning community that is bringing together like high school teachers, middle school teachers, and members from the community. Some of them who are considered scientists, right? So we have a group of beekeepers in Chicago. We also have some urban farmers. We also have Museum of Science and Industry that is coming together so that scientists can talk to scientists and see when that happens. The important part is figuring out how do we make people feel included in the scientific process. So we think that by sending students out into their own community and seeing how science is working with bees, right? They can feel like a scientist without even leaving their community. They don't have to be at Harvard, right?
What is your approach for, involving students in reading primary literature?
Reading primary literature. So it's funny, it's like classic papers, I think sometimes are easier for students to like grasp onto. As you read more current literature, I think that if you don't understand, you know, a lot of the background, a lot of the papers that are being referenced in the introduction, then you might not see, you might be able to read the paper and maybe understand what's a control, what isn't a control, what's the major finding, but not the importance of it. Right. And I think that if you don't have a student that can see how important the paper is or how important the paper is to the data, it's hard for them to engage with the material.
Okay, so you introduce them to the classic papers first?
Yeah.
One minute question in there. Yeah. Oh, so when you talked about using that Cas9, not unit, were you talking about CRISPR, or is this a different Cas10?
Yes, yes. So I think it's called CRISPR now. But I think at the time it was just enzyme that Cas9. But it is CRISPR inhibition, right? So where the Cas9 protein has that enzymatic pocket that's been mutated, and then it goes and binds to the DNA at the target site, but then just binds and doesn't allow transcription. So it's a little bit different going back to the genetic compensation, and that's why I don't know, like that, like maybe that's enough to cause that genetic compensation mechanism to like start up, but it is pretty well studied in fish that mutations, you know, in P11A might cause P11B to turn on. And actually, yeah. So you asked me about the mTOR. I'm sorry, the ATIC. Yeah. You asked me about ATIC. But what's interesting about P11 is if you inject a morpholino for P11A, right? You see P11B go up. Right? So there is like some compensation between those two as well, but we can talk about that.
We're running into class. Okay.